We've had a very stimulating 2 days of conversation, starting off talking about the different viruses and how they're impacted by their own subspecies classification, and then talking about the different approaches that are used to guide classification and the tools that have been developed both to support classification itself, but also to support the user utilizing the methods and utilizing the classification. Now, today we're going to continue on with the theme of looking at a few of the pathogens that are important from a subspecies standpoint, but also start to turn our perspective to the response. And so the response to a particular outbreak and what the different stakeholder communities would need, which includes NIH, CDC, and others. And so we'll hear from representatives from some of these agencies. So I'm going to introduce then Paul Eder from NIAID, who will moderate the session this morning. Paul, thank you.
Thank you all for participating in this third day of what's been a really, really valuable workshop for me. One of the first slides that I think the early presenters made in day one was this group of experts, either online or in person, and then some other people just generally interested in the topics. So I'm clearly in the latter because I'm not in this particular space, but I find all these discussions really fascinating. So I'm a diagnostician, and I'm interested in developing the tools to predict and detect disease earlier and earlier in outbreaks. So I have a great interest in this session because there's a public health focus, but from different perspectives. And the first perspective is from my colleague, Liliana Brown. She's the director of the Office of Genomics and Advanced Technologies inside NIAID, and she'll present on TBD. Oh, that's what makes it exciting. So, yeah, Liliana.
Hi, and I think Caden has my slides. Good morning, everybody, and want to echo Paul's thanks for everybody being here and staying, chugging along through these 2 long days. I know I feel like it should be Friday already, but it's been great. So I want to really, I've been thinking about it, of course, over the course of the last 2 days and even before that, and how I really think this is a critical aspect of responding and preparing for pandemics. I know it is very nerdy, but I do think this is very, very important. My slides are a little generic, but I hope that it will incentivize some discussion. And certainly I'm happy to talk here or via email afterwards as well. Can I have the next slide? Okay, so I thought I would just lay out the scenario of the case of NIAID's approach to pandemic preparedness. So we published a strategic plan for pandemic preparedness in 2021, and you can go and look at it. It's online, but it really centers around 3 major goals. One of them is to systematically characterize known and emerging threats, and that means really knowing as much as a body of knowledge and resources that we can develop for those particular threats. In the case for virus, and we're talking about, we want to be able to shorten the timelines between that original outbreak and the development of countermeasures, right? So we want to be able to deploy diagnostics, deploy therapeutics, and deploy vaccines in a much shorter timeline that we've had up to now. And last, as NIAID, we are a research body. We really want to be able to fill the existing gaps in research infrastructure, both clinical and preclinical, to develop much more of that knowledge base and respond much more quickly. So those are our 3 objectives in our strategic plan. And we've had a number of different programs here. You can see this detailed through, we deploy these through notices of funding opportunity, which are competed extramurally. And we have, of course, our intramural NIAID researchers who also contribute to the strategic plan with their own programs as well. But these are all mostly through our extramural funding opportunities to investigators such as yourself. And so we have programs dedicated to each of those 3 goals, and you see they are all responding to different aspects. Some of them do overlap multiple goals, so it's not like you can only do one or the other, right? Respond. And generally here at NIAID, we have had 2 major sort of philosophical approaches to this question of pandemic preparedness. Right? So we have those pathogens that we know pose a great threat. And for those, we call those our priority pathogens such as influenza, such as coronavirus, where we know they have they have caused pandemics in the past, and we want to develop as many resources as possible to rapidly respond in case of a new emerging virus. So that's our priority pathogen. We also have our other approach, which is called the prototype pathogen, in which case, as the name suggests, we're using a prototype, and this is where I think is really relevant what we're doing here, to say, can we develop enough of a knowledge base, enough resources that we can then apply what we learned from that prototype to anything that may be coming that is completely new and is not going to be necessarily what we studied in the laboratory, right? So this is where you see how similar is this prototype to what actually is emerging, right? And then how easily can we make inferences from what we did in our prototype to that particular outbreak that we're detecting. So we have those two prongs which are addressed through our different programs. I wanted to highlight the different programs briefly for all of you to understand what it is that we're doing in all of the different fronts. So this is an example of the prototype approach, right? Where we had a SARS-1 outbreak. We developed a number of resources, candidate vaccine. We were able to improve on that candidate vaccine when there was a MERS outbreak about 10 years after that. And then 10 years after that, we had SARS-2, in which case we knew how to stabilize the glycoprotein. What were the immunogenic components of that glycoprotein to make the most efficient vaccine and deploy it in the shortest timeline that we've had so far for vaccines. So this is the idea behind the prototype approach, that you have something that is really prototypical of that class of viruses that then you can very rapidly put that in action in the case of an outbreak, of an emerging outbreak. I've been saying prototype, priority. Get them confused. We should use different names. The prototype pathogen really is, as I mentioned, developing that body of knowledge about this prototype so that then we can project that information into anything that is emerging and make generalizable approaches of how it might work for those viral families. So our approach for the prototype, some of you participated in this process, which was really getting advice from experts in the field regarding what are those pathogens that pose the highest risk of morbidity and mortality in humans that may cause pandemics. And then we categorize these according to how many resources and what's the body of knowledge that we had about those particular pathogens. And so you see here our classification system for these into 4 different classes, those that had high potential for pandemics, but also had high number of resources, a lot of information that we know about them. We had those that had high potential for pandemics, but maybe moderate or many fewer resources. And then on the other side, we had moderate pandemic potential, high resources or low resources, according to the pathogen type. Here at NIAID, we decided to focus a lot of our effort on those pathogens in the purple rectangle, which are the ones that had high pandemic potential, but not as many resources, and certainly not as much known for those particular families. And so we see here, and as I detail some of the programs, you'll see that most of them are addressing these particular families. And I feel inadequate talking to actual viral classification experts about the different families. And Peter can tell us again whether they're families or species or orders or whichever way we want to talk about. This is how we approached it. Okay, at least we use those classes that we have right now. And you can read all about this both in a publication that was done in, I think this is 2022, very early. And since then, we've also published a number of supplements, and a supplement that detailed each one of these particular families, sort of catalogs what it is, the knowledge and resources that we have for them. So I invite you to look at those as well. And so in order to address sort of the systematic characterization of priority and prototype pathogens, we've deployed, in this case, the Centers for Excellence in Influenza Research, as well as our Centers for Research in Emerging Infectious Diseases. So we've heard about the SEER from, I think Derek talked about it, Indresh talked about it as well. This is a network of investigators that are mostly funded in the United States with global partners that do a lot of surveillance and respond, generate resources And here's just an example of something that they're doing currently for the H5N1 outbreak that has been ongoing for now almost 2 years, I think. So they'll do animal surveillance, they'll do human cohorts, they'll do seroprevalence, they'll develop reagents, they'll develop viral stocks, highly characterized viral stocks of those particular pathogens. They'll look at how does isolate behave in molecular assays? How does it behave in animals? Does it transmit differently than other pathogens that we know than other isolates? So here, for example, we know we've been talking about it. Which is the isolate that we choose? How do we choose that isolate in order to do all of these assays, all of these experiments? Is it the first one that emerged? Is it something that is later on? When and what is the right isolate that will give us the best knowledge so that we can properly respond? And we, as I mentioned, also fund the Centers for Research on Emerging Infectious Diseases, who have a similar mandate, but not on influenza, but rather all the other pathogens. And again, they're trying to understand the risks of viral spillover, develop resources, make surveys. This one, sorry, I should mention, it is an open funding announcement right now. You can link to it. My slides will be available. So if this is something you're interested, they are poised globally with investigators that are doing all of the field research, human research around the planet. And you see as well, I don't know if you can read it, I know it's small letters, but the types of pathogens they're addressing in each of the continents and each of the geographies that are relevant to that particular human population and risk of zoonotic infections, but also address that pandemic preparedness plan that I was discussing earlier. In the space of vaccines and monoclonals, we have invested into our revamp network, which is using the prototype approach, the prototype pathogen approach. So they have the goal of this program is to develop those vaccine candidates really to a point where they can then be rapidly deployed in case, or the generalizable approach could be used in case of a novel outbreak. Right. So we're getting the vaccine candidates to a point where we understand enough about them that we say, this is the good candidate that we can possibly use in the future, or for monoclonal antibodies. And again, they continue to develop the knowledge and reagents and those animal models and those cellular models that are so critical during that response, certainly to understand immune correlates of protection as well. So in this case, these were the target viral families for that program from the Revamp Network. And I believe the notice of funding opportunity was also very specific on the actual viruses that were expected to be used. The applicants could, of course, propose their own viruses according to their own expertise and their own experience with any viruses. But these were sort of what was chosen as the best possible prototypes for that family of viruses, or that class of, however you guys want to describe them. We have also invested into programs to address the need for more broadly vaccines that protect against a broader group of pathogens in that particular class. So, for example, we've invested into Project NextGen, which was really trying to develop a broader coronavirus vaccine, as well as the program for the pan-coronavirus, which was really trying to address the wider diversity within the sarbecovirus family. And we have also invested into doing similarly for the influenza vaccines, really trying to get more broadly protective influenza that are what we call variant-proof, if we want to think about it that way, improved for seasonal influenza, and make, I don't know if it's here, I might have taken it out, the universal influenza vaccine is sort of a goal that has longer protective, it has durability of protection as well as being broader across a larger group of influenza isolates. And we have also invested into antiviral development, and in this case, again, we use the prototype pathogen approach. So we funded this work to do better antivirals against coronavirus, but again, also against 7 other viral families. and with the intent of developing antivirals that could be also deployed in case of a novel pathogen that is within the same, similar to what we're studying in the program. And these are the viral families that were studied within the AVID program. And you can see here, most of the antivirals are sort of targeting some of the components of the virus that might not evolve as quickly as we see those that are the target for immune protection. And so I wanted also, I've laid out a little bit of how NIAID has structured our response through these large programs to address the need for monoclonals, for vaccines, for antivirals, and how we're really addressing the concept of knowing those viruses that we know pose a high risk, so those priority viruses, but also the ones that might emerge that we don't know, but are maybe similar to other ones that we are addressing. So I also wanted to highlight some of the work that we did during the course of the pandemic in responding specifically to the variants as they were emerging during the active ongoing pandemic. So here we had working groups, including the SAVE, which I'll show here, TRACE, which again, some of you participated in activities with TRACE. That was critical interaction with NCBI. And then a little example of how this is going on within the SEER network to establish a risk analysis pipeline for influenza research as well. So that's what the RAP is. And so I thought I would just show you, this is directly from the CDC website. This was a risk assessment that they did early on in one of the early variants. I don't know which one I chose, but I just clicked on one of them. But I wanted to show you what it is that at least in terms of developing the knowledge that we need in order to understand the risk that a particular variant poses. So we want to understand the epidemiology of that particular variant. This is different, right? So we're looking at human cases. Is the number of human cases significantly different that we've seen for our other variants? Is it spread in the different parts of the world? And does it have, so I'll go through each one of them. Does the particular variant cause a disease that has a phenotypic difference from other variants that existed before, right? So the severity of the disease is something that we need to understand. Does it have a different transmission pattern? Do we see that it's now, instead of potentially transmitting from the lower airway into the upper, or it's more into the upper airway like we saw with Omicron, that dramatically changed the progress of the pandemic? This is changing between hosts, for example, would be something that you want to understand. Can we see, in part of the epidemiology, CDC deployed the wastewater detection program, which I think has been critical really to understand how the virus is spreading across a community. But this really contributes to the epidemiological data of how the variant is transmitting differently, and also gives us an understanding earlier compared to hospital cases, because we're able to detect variants sooner in the wastewater than we would see in the hospital setting. And we use, of course, viral genomics, right? Because that will tell us how different and how diverse that population of viruses is. And we want to see whether this particular variant has immune escape or it has impacts on the immunity that would be relevant to updating a vaccine or updating monoclonal antibodies that would be protective. And last, of course, any therapeutic or diagnostic effects of that variant on the efficacy of therapeutics or diagnostics is critical. And we saw, for example, many of the diagnostic tests had to be dropped because they wouldn't be able to detect novel variants as they were emerging. So this is the knowledge that we need to really assess the risk of a new variant. And I think I showed you how some of our programs would be able to develop a lot of the knowledge base that would be needed to make that kind of assessment. And we saw, certainly from the CDC, another method of classification into what is the risk that every single one of these variants poses. And according to criteria developed from these experiments, we would be able to then categorize them into each of those. So here at NIAID, early in 2021, sort of in response to the emerging variants, we decided that we needed to have a more coordinated effort to really address the emergence of variants. So we established a SAVE consortium. I don't know what we call it. Is it consortium, Derek? What's the word that we use? But it really is the SARS-CoV-2 assessment of viral evolution. And it was really trying to save us from the pandemic. So it was a play on words on that. But it really was intended to coordinate the efforts across all of our different laboratories that were doing all the necessary work in collecting that critical information from the last slide to feed into making the public health decisions that were so critical during the pandemic. So in here we had a group, the structure it was based on 3 major working groups. We continue to have biweekly meetings with these 3 different working groups, and then one in which all of us get together and discuss the status of the pandemic. So this has an early detection and analysis working group. Elodie, I think, is our chair for that group. And so if you have questions, she'll be more than happy to talk to you about it. But here we really is where we coordinate the knowledge that all of us are generating, both for the sequencing from the trees, the phylogenies, the diversity of the pathogens. All of that feeds into this group. And this group then makes an assessment and advises the rest of the working groups regarding which are the variants that we should be evaluating in those molecular assays and further downstream into those animals. So the in vitro characterization group is the one that's deploying all of the neutralization, all of the binding, all of the sort of things that we can do in very high throughput to give that knowledge to the then in vivo group that then will have to bottle. We have a severe bottleneck there in how many variants we can test in animals. But only for that, then we get into the animals and get that information about how is this variant different than other variants, and do we need to then really look at it more closely and make other types of public health decisions. So all of that feeds into this variant risk assessment program. And here I don't want to go into a lot of details. This is Eric Stemmy's slide regarding the process that how we use and deployed SAVE in response to that Omicron outbreak late in 2022, right? Dates are all confused now. But you see within days of having the sequence being released by our colleagues in South Africa, then we were able to make those constructs and get those into the assays and get them into animals so that we could understand the real threat of each of these different variants. And it is a very critical engine to make those kind of informed decisions of how the variants are causing risks. And he mentioned we're doing something similar through the SEER network, and we are developing a similar approach and with similar knowledge base to try to make an assessment and inform that can inform the risk process decision, the decision of public health responses globally and in the United States. And here, so I just want to end on this slide, which is this is all great, but it is also reactionary. And I wanted to say, ultimately, what we want to use the information that we have here and all of the knowledge that we've discussed is really to try to make better predictions of what may happen. So the CDC has developed, for example, this Nowcast. I hope we hear a little bit about it, which is a future prediction tool, because the knowledge, even though it's telling us what the situation of a pandemic is today, all of the data that they use for that Nowcast model is actually about 3 weeks old. 3 to 4 weeks old, right? So it is a predictor of the future, but it's really telling us what's the situation based on how the virus behaves and the progression of the pandemic, what may be the constitution of the variants at this moment. So we need to continue to improve on those modeling capabilities to make us be able to react sooner or even preemptively to any possibility of variants. But also on the other side, from our molecular assays, our in vitro, in vivo assays, be able to make better predictions of what's the impact of those mutations even before they emerge, right? So we can build models that'll tell us whenever we do see this in the population, we know when we need to be worried. So this is where we have this new modeling approaches that are telling us when these particular mutations from day 0, right? We have the sequence, and from day 0, we can already say, we expect that these will have these particular effects on immune escape, on vaccine, on therapeutics, monoclonals, et cetera, and diagnostics. So that's where I hope we can build this information, is for that future prediction. And with that, I'll end and I don't know if there's questions about this later, right? So thank you very much.
So I think Liliana really gave an awesome description of the foundation of research that we really put a large effort in at NIH for these outbreaks. Another perspective on what's going on in outbreaks is in the area of preparedness and response. And it's work with one of our sister agencies in HHS, and that's the CDC. So I personally work with CDC scientists on a lot of readiness tools in the diagnostic space. Here we're going to hear from Andrew Beck in the National Center for Immunization and Respiratory Diseases on modeling and other aspects of measles outbreaks. Andrew?
Good morning, everyone. Does this podium go up? Is there a button? I don't want to present hunched over like this, but I'll do my best. So my name's Andrew Beck. I'm from US CDC in Atlanta. I'm a research virologist in the viral vaccine preventable diseases branch. Our stories across a number of pathogens for which licensed vaccines are available. So I'm going to be discussing measles virus this morning because it's really too much to cover the entire federal perspective. So this is here's my disclaimer. These are my remarks only. These are not the official position of the agency necessarily. Okay, so I bring up measles virus today because it's a very, very, very timely issue. This is just a clip from a recent MMWR reflecting on a set of cases that are being addressed on an emergency response basis in Chicago presently. And I will be a little bit later in the talk discussing Operation Allies Welcome. We published a little bit on this in 2023, which referred to our emergency response to measles cases that were observed in the Afghan evacuee population following the fall of Kabul in 2021. So I'm going to discuss the virus a little bit because the diversity landscape, I think, is a unique one for this particular pathogen. This is a much different situation from what we find with SARS-CoV-2, for example. And I'll give a little bit of the biology. So here's the obligatory genome organization slide. I only show this to demonstrate that that typical molecular surveillance of measles is done with partial genomes. And there's a number of reasons for this, cost being one of them. But so if you look over towards the 3' end of the genome, this N450 segment, this is the typical window that is acquired during surveillance sequencing. And this is done universally across all surveillance networks. The virus is monotypic, and this is canonical information. And it's also known to have pretty low substitution rates. So this graph that you see over on the right is from a review by Richard Klemper. And if you look on there, this is a meta-analysis of reported substitution rates for the virus. And you can see, well, measles virus is over here all the way on the low end of the spectrum. So this is considered to be a slowly mutating virus. Our current typing system was proposed in about 30 years ago, and it's based on the nucleoprotein. Our current genotyping uses 8 clades. They're assigned letters, so A through H, with a numeric designation of genotype. So there's 24 of these in total that have been described. So there's a letter and a number. So D8, B3, D4. And there are standard sequences that are available to perform typing if you wish to have those. We also have a concept of what's called a named strain. A named strain is a persistently circulating strain of the virus that is observed in the surveillance data over numbers of years, and that's human curated. We also have a concept of a distinct sequence ID. So since this N450 segment is so short, we can actually assign an independent integer to a unique sequence in the database, and we track them that way. And so this N450 is universally collected, and we perform extensive research on sequencing techniques to get the whole genomes when they're useful. So the current nomenclature is, as you see here, we're able to communicate whether or not the virus came from a clinical case or it came from a cell culture isolate. We have some geography. We have an epidemiologic week, a year, a multiplicity integer. So this is if there are multiple cases within this time period, and we have a genotype at the end. So there is a greater need, and I'll show you why in a minute, but there's a greater need for more epidemiologically meaningful nomenclature, and that's an active piece of our research, actually. So this issue of low diversity is a consequence of a number of things, and that's what these next few slides are going to be about. So the virus has a restriction baked into the cake because it has to negotiate multiple receptors in order to productively infect the host. So this is the one that you really want to pay attention to. So upon entry, the virus is picked up by lymphocytes, and it predominantly infects lymphocytes. But it's picked up by the SLAM receptor, which is a lymphocytic receptor. And in order to exit from the host, it has to negotiate with the Nectin-4 receptor in the basal surface of the respiratory epithelium. So the hemagglutinin has to negotiate. So this idea that the hemagglutinin has to negotiate these two receptors implies a restriction on the mutational profile of the virus, writ whole. And epidemiologically speaking, we're also in a decreasing diversity situation. So this is a set of genotype surveillance data that was published. These are sequences that were submitted to our central surveillance database between 2016 and 2018. And you can see that during this period, there were 6 genotypes that were reported. The situation is different now. In the last few years, present day, we really only see 2 of these genotypes out of 24 being reported. So the other thing that's at play here is that there's a thread in the literature about this issue of the quasi-species profile of the virus being constrained, and that there are considerable structural constraints, especially on the hemagglutinin protein, with respect to the language that's used in the literature is one of co-dominant neutralizing epitopes. So there are multiple regions of this hemagglutinin which offer neutralization. And so from a quasi-species perspective, there's really only been one major quasi-species profile that's been described in the literature. This is a quasi-species that involves this editing site that causes the difference between the expression of the phosphoprotein and the V protein here. And so this quasi-species alternates in its appearance between lymphocyte cells and epithelial cells. So you can see that there's this pattern here that this virus is quite happy with its niche, and it's a very specific niche that it occupies. So epidemiologically speaking, there's a chief interest in the United States, anyway, is the maintenance of measles elimination. And that has a formal definition. So this means that the absence of endemic measles transmission in a defined geographical area for at least 12 months in the presence of a surveillance system that has been verified to perform well. Our chief concern is to maintain this. So measles has been eliminated in the US since 2000. So we wish to maintain this elimination status by tracking a virus that is most contagious, structurally constrained, genetically stable, in an environment of decreasing diversity of circulating sequences, while bringing to bear a large surveillance network, all of the members of which are able to bring various levels of resources to bear. So you can see that this lack of diversity becomes a problem over time. There's a classification issue, as you'll see in a second. And so baked into this idea of elimination verification is our chief interest, which is the discrimination of transmission chains and the interruption of transmission continuity. So this is an incredibly difficult problem as genetic homogeneity becomes more of an issue epidemiologically speaking in our surveillance data sets. So you can see that these two transmission chains could be functionally equal in the presence of homogeneous sequences. So this is a real issue. This is just some internal data where I wish to demonstrate to you that our understanding of measles classification is in a little bit of a transition period because we're increasingly using whole genomes, but that's not really a practicality in our surveillance networks right now. So on a research basis, we find ourselves in this position where the trees are actually the trees are rearranging a little bit once we add the whole genomes into the mix. So these colors reflect our distinct sequences, and we frequently find distinct sequences mixed up in monophyletic groups when you run Bayesian analysis, for example. So integration of sequence data and outbreak response. I'm going to discuss very briefly part of this publication that we released in 2023, where we we looked at measles transmission during Operation Allies Welcome from multiple perspectives. So there's really two stories in this paper. One of them is the modeling, and one of them is the genetics. I'm going to be discussing the genetics, but there is a broader story here that we don't have time to discuss this morning. And so you may remember that in late 2021, following the fall of Kabul, there was 80,000 evacuees from Afghanistan that were brought to the U.S., and they were temporarily housed on military bases for the most part to await further processing and quarantine and et cetera. This is the epicurve of the 47 measles cases that were observed during this period of time. This gray bar is vaccine uptake during the mass vaccination that was ordered during that period of time. And you can see that this is so the big takeaway from this diagram of this is that this is in fact a very short period of time. So August to October. So we're talking 2 or 3 months really is the temporal scale upon which we're working here. We were fortunate in that we were able to get whole genomes for about 43 of these cases. And there was and in order to put this complete story together, you can see that we've got this, we get very flat trees for these viruses at certain times. And this is one of them, even considering that there were whole genomes being used. Over here on the right, there are a number of pertinent epidemiologic variables that were collected to support this data set. But the one that I want to direct your attention to that I think is really the most important for discussion here is the importation status. So our epidemiologists are not only classifying the case linkages to the extent that they're able, they're also classifying whether or not a case is imported into the U.S. borders. And so one of the big takeaways here, and I want you to look at this flat clade up here. There's this one, and I want you to look at how complicated the importation pattern is across this flat clade. This is the chief problem with short outbreaks of measles. We get these flat trees, and sometimes we get these and in this case, we got a very complicated pattern of importation. So according to the epidemiologists, they're proposing about 6 discrete infection clusters here, and there's a number of these infection clusters overlap. So we're seeing repeated introductions of similar or identical viruses. One of the takeaways from this paper, and we wrote this in discussion, is that this is one of those situations where we're reaching the limits of resolution in scenarios of complex importation of genetically similar sequences. This is really, the takeaway from doing a piece of work like this are really 2 or 3 things. So one of them is that a piece of work like this forces the geneticists and the epidemiologists to get in the same room and collectively interpret the data together. Right. This is kind of an art, right? Because when you take 2 disciplines and you sit them in the same room, there's a language barrier that has to be crossed. But I think it was successful in this juncture. And the payoffs are that we're able to cross-validate our own data sets and ultimately get a more complete reconstruction of the infectious process, I think. And so this issue of revising measles classification for elimination context allows us to reflect on a few of the major needs. And one of them is this issue of heuristics. And I use that word as a shorthand. And here's what I mean. So in our large international surveillance networks, we cannot ask our partner laboratories to do computationally intensive things all the time. There are some labs who have the resources to run beast trees for weeks on end, and some labs that do not. And so for those of you who are working on base tree assignment methods, we've got our eye on your softwares because we're incredibly interested in being able to allow or assist our partner labs in performing classification tasks without having to have them build their own clusters and run beast models, for example. We are incredibly interested in methods that allow us to use sparse and multifactorial data. So Tavis, this is something that you reflected on a few days ago. And this is one of those cases in which it's a huge need. So when we find the limits of resolution for the genetic data, but we have all this other epidemiologic data to bring to bear, frequently, well, either one set or the other is a little bit sparse, but there's a greater quantitative picture to be made there. And so those of you who work on those techniques I'd love to talk to you. We also need validated software models and parameters. So I think we can call this the grandmother test, I think. So if you write a piece of software and you would feel comfortable giving health advice to your grandmother based on the results that the software provides, that's what I'm talking about here. We want software with well-described behavior that's well-tested, well-integrated, and well-maintained. These are when you're giving health advice to lots and lots of people simultaneously, this is a big deal. We're during measles classification schemes in general. This is ongoing on our laboratory. Our standard data is heavily controlled and really reliable, I think. And so whatever additional information gets used as this scheme arises will be epidemiologically meaningful to approximate transmission lineages, which is really our chief interest. So anyway, thank you for your attention and look forward to your questions later.
Next, we'd like to hear a more global perspective from Rosamund Lewis. She'll talk about mpox.
And I am coming to you from Kinshasa in the Democratic Republic of the Congo, although I cannot blame the country. It's definitely my computer. I'll take it in for service when I get back. So now you're in for something completely different. So sit back and relax. I'm a public health physician. I'm not a virologist. I'm going to take you on a whistle-stop tour of the global mpox outbreak and try and bring some of the viral evolution elements into it for you. So if I can also, there we go. All right. So my first slide here is just to illustrate briefly where we are in the global mpox outbreak, which you may recall was a public health emergency of international concern in 2022 and 2023. This is data from February. Our data from March is coming in now, and we'll be having that shortly. So what you see on the top left is the number of cases of laboratory confirmed cases reported globally since 2022 and up to the end of February of this year. So over 2 years now, of almost 95,000 laboratory confirmed cases. I insist on that because we'll see why later. Just 181 deaths have been reported during this time among laboratory confirmed cases, but the epidemic did reach 117 countries. If you look on the right, what you see is the reporting from February of this year, where notwithstanding the decrease in number of cases, which you'll see in a moment, in the last month, in February, there were still over 700 cases reported from 29 countries around the world and 2 additional deaths among confirmed cases. So what you see on the left in this slide is a beautiful epi curve. Epidemiologists love this kind of stuff, where a dramatic upswing in the number of cases, control measures are implemented. The outbreak may also naturally, to some extent, subside. And what we see is a very, very long tail on this outbreak. And at the end of that tail, around mid-2023, August, September 2023, we start seeing a little uptick in cases again, but at a much lower level. So what you see on the right is the number of cases reported by WHO region. We have 6 regions, 6 regional offices with the World Health Organization, and you see them there on the bottom. Again, you see an evolution. The colors are changing over time. And so even in September of last year, there were outbreaks in the Western Pacific region, and they're now seeding again to more cases in the Americas, Europe, and Africa. This gives you a bit more of that flavor. You have on the top right region of the Americas, so that's all of North, Central, and South America, and bottom left, the European region. Those are very similar curves. These were the areas first hit by this outbreak. The others look a bit different. The Western Pacific and the Southeast Asian regions experienced their outbreak in the end of late 2023, and subsiding now. Whereas you can see the top left, the African region looks very different. The Eastern Mediterranean region did report cases in the early phases of the outbreak, but many countries have ceased reporting around the world, actually, that is, which is a problem. This is just to highlight basically the deaths, and we start getting into a little bit. This is, again, it's still among the confirmed cases, but nonetheless, we start seeing differences by geography, which can be attributed to quite a number of reasons. But first, let's take a look so the deaths, the epi curve is following, and the bottom left is following more or less the trajectory of the global case count. And on the right, what you're seeing is case fatality ratios, which is the proportion of cases over the proportion of deaths over the number of cases reported. And we see that globally, we've had a very, very low case fatality ratio. Of course, some people have unfortunately died, but overall, globally, it's been low. But compare the European region at 0.03% to the African region at approaching 1% overall, even among the confirmed cases. And you see we're having an order of magnitude at least difference. So this may be due to, in part, in surveillance systems and detection of cases, access to and quality of health services, presence of comorbidities such as uncontrolled or untreated HIV infection. But also we have our monkeypox virus clade, one now, we'll come to this in a moment, 1A, 2A, 2B, and lineage differences and gene deletions. We'll come back to that in a moment. This is again from the global data. So the number of cases are relatively low at 2,700 cases reported due to challenges in accessing confirmatory testing. But nonetheless, what we see here in the epi curve on the left, the yellow represents Nigeria, which, as we know, is in many ways the origin of the outbreak, which began in Nigeria in 2017. This data only reflects since 2022 as for the rest of the global data. But nonetheless, Nigeria experienced a significant outbreak. And what they have there is what was called West African clade is now called clade 2. But that has been superseded now in 2023-24 by more case confirmations from the Democratic Republic of the Congo. So we're still talking mpox, we're still talking monkeypox virus, but we're talking different geographic locations and different strains. What you see on the right is a typical age pyramid for cases in Africa, which is dramatically different from what was seen globally. In Africa, still the majority of cases are in children under 15, whereas globally it was adults, men primarily. So where were we last August? The Director General of the World Health Organization, under the International Health Regulations, issued standing recommendations following consultations, many consultations of experts around the world. And these are some of the recommendations that were come up with. I've highlighted in red for you the ones that are relevant to testing and specifically, and therefore the nature of the virus itself. So strengthen and sustain testing and surveillance capacity and ensure that new cases are nationally notified. Invest in research to better understand mpox disease and transmission patterns and develop improved vaccines, tests, and treatments, and work towards equitable access to safe, effective, and quality assured vaccines, tests, and treatments. This applies to all member states, which is virtually every country in the world. So let's move now to what's happening right now in the Democratic Republic of the Congo. What you see on the upper right is actually an epi curve that spans decades. So we're not looking at one over the last year or the last few months. We're looking at, on the left, going all the way back to 1996. Zaire, the Democratic Republic of the Congo, was the location where the monkeypox virus was first found in a human, a child, a 9-month-old child. And so that was the first documented human case of mpox 25 years prior to the beginning of this particular epi curve. But as you can see, what we're seeing now is a steady increase over a long period of time of the number of cases reported. The country uses a clinical case definition, which describes classic mpox, which is very similar to smallpox, and therefore these case reports, whereas of course many of them may in fact not be mpox, they do fit the national case definition. They're not just alerts of fever and rash, for example, which might be due to chickenpox or measles. And so there are other causes of rash and fever, of course, as you know, but this is clearly an increase, significant and notable increase in the reported number of cases over time. And so You can see at the height of that curve at 2023, there was already a significant increase over the preceding couple of years, which were pandemic years, but even over 2020 and the years before that. What has also changed in this period of time, you see some text at the bottom, is that not only has the number of cases gone up, but the case fatality ratio, the death rate, if you will, of among cases has also climbed significantly. So from 2021 at 2.7%, it is now nationally in the first quarter of this year, 2024, at 6.5%. So WHO organized a joint mission with the Ministry of Health. This was a comprehensive mission which included different departments of the Ministry of Health, and including the National HIV/AIDS Control Program, due to the nature of the outbreak globally and due to the fact that sexual transmission have been documented in the DRC for the first time in 2023. So this was a new feature for Central Africa with Clade 1, but it was not new globally, but we needed to kind of integrate the skill sets of the emergency program and the HIV/AIDS control program. So this mission visited 4 different sites in remote parts of the country to really provide a description, and a report of that is available online. Unfortunately, that's in French at the moment, but I believe some translations are floating around. This is, again, illustrating what we're seeing now in 2024. This is already a few weeks ago. I apologize, I don't have the last month's data on here. But in the blue line is the cases and deaths from 2022. The red line is from last year. And you can see that in 2024, reported number of cases are rising and also falling, because in the last month, there has been, in the most affected area, there has been a mission to strengthen response and patient care. And there is an understanding that that at least has contributed in some measure to the decline in case fatality in that part of the country, which you'll see in a moment. So I showed you earlier, let me go back to it. So this map of the Democratic Republic of the Congo, which is a vast country, illustrates in dark blue the most endemic provinces of the country. And on the left is Equateur province, and that is also home to some outbreaks of Ebola and other such things. But it is the most affected province in the country, but there are others. As you can see that the quite a few provinces have now been affected. 23 out of 26 of the provinces were affected in last year, and many of those have been newly affected. Those are in lighter blue or even in white have been affected since then. So the second one that's circled is South Kivu. This was a previously unaffected province. It had never reported mpox before. When it began reporting an outbreak of mpox among sex workers and other members of the community, and this was very notable. 30 or 40% of cases are documented among professional sex workers in a mining area where there is a lot of cross-border travel, both of the sex workers as well as of professionals in the mining industry. So it is a bit of a hotspot that we are concerned about on the east side of the country in insecure areas, but where there's clearly an outbreak of sexual transmission, which again is new for this part of this clade in this part of the world. On the other side of the country, in Equateur, where I just showed you, there is this is an epicurve for that place, which shows you again that there was quite a significant increase in number of cases in 2023, which subsided a bit. We don't know whether that's artifact and surveillance dropping off, or whether it is true decrease in number of cases. And then again, picking up again in 2024. There's no doubt that there's clearly a significant outbreak still ongoing in that part of the country. And again, even within the province, focused geographically within certain areas in health zones, and even within the health zones, focused in geographic areas, which are called health areas. The more you get down to the local level, the more you see certain features appearing, such as a very high case fatality ratio. So we're looking at more than 1 in 10 cases are in fact dying in this part of the world of mpox. These are reported cases meeting the national case definition. They're not all laboratory confirmed. There is an ongoing concurrent measles and mpox outbreak, both of which have been confirmed in Lutumbe province. So that illustrates some of the complexities that are there. Also illustrates the very high incidence of confirmed mpox and suspected mpox in those regions, the high case fatality ratio. Just a little historical note on the bottom left of this slide is that for smallpox, prior to eradication in 1980, This country reported in 1963 its peak. The maximum number of cases it ever reported was in 63. And that was a peak of just, if I may say, just 5,523 cases and 710 deaths. The case fatality ratio for smallpox was 12.8%. And that, relevant for your group, was two different strains, variola major and variola minor. And the CFR was 16% for variola major in the country. And the 3/4 of the cases were variola major and 2.9% for variola minor. So we're seeing a similar picture to what we're seeing now with clade 1 versus clade 2. We were already seeing it for smallpox with variola major, variola minor. But also the order of magnitude of the number of cases reported is significant in comparison to what we're seeing today. This is another interesting feature. Bolomba Equateur, you've seen already in the map. So that is the endemic area, the top left age pyramid. I've already mentioned the vast majority of cases are in children under 15, and most of those are in fact evenly split between children under 5, which in Bolomba is even higher. So very high prevalence among children, incidence among children under 5. And on the right, we see a very, so the next one, South Kivu, we see a very different picture. So that is the picture of the demographic pyramid of the outbreak among sex workers. Few children are affected in households, but primarily among young adolescents and young adults. And this is the first demographic pyramid I'd ever seen where there were more women affected than men. So clearly an emerging public health problem for both women and men, and representing a risk to neighboring countries because it's in a border area. So in summary, we have a rising number of cases and deaths later. There's a bit inconsistency here. Later, I say 10%, but really it's between 10 and 13% that we're seeing of laboratory confirmation in 2024, which is increasing. The country's improving access to diagnostics, and most of the laboratory tested cases ranging of above 60% test positive. In endemic areas, it can be over 75% or over 95% of tested cases are in fact positive for monkeypox virus. We're also seeing for the first time ever, at least confirming for the first time ever in this country, co-infections with HIV, syphilis, and other sexually transmitted infections, which mirrors what happened in the global outbreak. I mentioned the geographic expansion in mining areas and border areas, and the sexual transmission is affecting not only sex workers, obviously, but also heterosexual transmission in the household, and even an anecdote of a health worker being infected at work and exposing his female partner at home. So clearly sexual transmission is a new feature of mpox, whether it's globally or locally. And we're very concerned about the rising case fatality ratio, which is affecting mostly children. I guess you're not used to seeing these kinds of photographs in your meetings, but this is what we do in public health. Very concerning pictures about systemic infections, sexually transmitted infections. And not to forget the origin, monkeypox virus is an orthopox virus, which is also a zoonotic infection. And so much work still needs to be done to understand how people are exposed and infected in contact with the forest or bushing in markets or other traditional practices. So this is kind of where we landed at the end of last year. Some of that data is more recent. I've showed you data from the first quarter, but at the World Health Organization, this was deemed to be a situation which merited continuing the grade 2 protracted emergency, which is different from the public health emergency of international concern under the IHR. The WHO has its own internal grading system. And so mpox global outbreak, including the new emerging situation in Africa, is considered to be a grade 2 protracted emergency. And this illustrates approaches to risk assessment that we use: risk for human health, risk of the event spreading, risk of insufficient control capacities, and overall confidence in the available information and overall risk. So let's move on to something which is more in line with the work that you're doing so far, just to illustrate geographically on the world map the different clades. So I've mentioned that clade 1 is Central African clade. Well, I'll mention in a slide or two that it was renamed during the course of 2022. And the red clearly illustrates that these are the areas where clade 1 has been documented. It's not been documented anywhere else in the world. Clade 2A is West African clade. And the outbreak in the United States of America occurred in 2003. It was linked to infected pet prairie dogs that had been co-housed with giant Gambian rats. during transit in importation to the U.S. And so the cases that occurred in the U.S. in 2003 were of clade 2A, and only one other human case of clade 2A has ever been documented. And then the blue is, of course, the clade 2B, which is the global outbreak, which has circulated the world, including Africa. Probably some mixed outbreaks in some places. This is what the map looks like. For the phylogenetic tree, I had 3 for you. One is from prior to the global outbreak. Sorry, that is meant to say clades 1 and 2, not just clade 1. And so you have the countries on the bottom prior to the global outbreak where these specimens were detected and sequenced. And then you see the clade 1, clade 2A I've just mentioned from the global map, and clade 2B from the global outbreak, which actually began in Nigeria with their outbreak in 2017. The Central African clade outbreaks continue sporadically in several countries: Cameroon, Central African Republic, Congo across the river from Democratic Republic of the Congo, and in West Africa, Liberia, Gabon, Nigeria. And it's really has been a very sporadic disease until recent times. Cameroon is the only country to have so far ever reported both clades. So this is the phylogeny during the global outbreak. We're definitely seeing here more specimens of clade 2B. There was a lot of sequencing, thanks to all of you who participated in that. It's really helpful to see where the outbreak is going. This is one of the major benefits of your work as well in this area. So as I mentioned, clades have been renamed. Central African clade is now clade 1. West African clade is clade 2. But clade 2 has been subclassified into 2A and 2B. And the global clade 2B outbreak was linked almost exclusively to sexual transmission, but we're now seeing that that's a feature of clade 1 as well. In addition, and what I've not mentioned yet, is new outbreak in Sudan. So there was an outbreak in Sudan in 2005. It was in what is now South Sudan, Unity State. And the 2 clades, I'll show you those in a moment, but more clearly than where they are here, the 2 strains, because there was a new outbreak in 2022, probably due to the greatly enhanced surveillance during the global outbreak. Hundreds of children were affected in refugee camps. Again, a completely different manifestation of the epidemiology, not so much of the disease itself. But that was found to be clade 1, but again, different from the clade 1 that is in Central Africa. So that's a new feature, linking these 2 outbreaks in East Africa, in South Sudan and Sudan, via the sequences that were done for those specimens in 2 different international reference labs, and over obviously a 17-year period. This is now the genomic epidemiology of clade 1 that we're seeing, actually globally and across time. But the 2 features are the one that I've just mentioned around Sudan, where these two sequences of 2005 and 2022, and then Kamituga, South Kivu, where there is the ongoing outbreak, which has not been stopped, ongoing outbreak of mpox, primarily via sexual transmission. And that, sorry, the arrow's in the wrong place on this slide, but there you see the number of sequences in pink, showing that they are somewhat different from the other clade 1 sequences. So we have divergent lineages from most of the clade 1, and we have to also say that a lot of the clade 1 sequences, even from DRC, have not been put in the public domain yet, and that is something we are working on with the country to publish those as soon as possible. But nonetheless, we're still seeing divergent lineages for the sequences from Sudan and the new clade 1 cluster of South Kivu in 2024, which has already been published by researchers looking at that on a couple of papers already so far. So in South Kivu, what have they demonstrated? And this has been confirmed in the National Reference Lab at the National Biomedical Research Institute in Kinshasa, where I am now, is that there are gene deletions in this new strain from South Kivu. And the gene deletion is specifically that the CDC recommended clade 1 specific PCR target, therefore leading to potential or actual, it's been confirmed, diagnostic failure if relying on clade-specific tests only. Whereas in the DRC, they have been using the CDC protocol of non-variola virus, orthopox virus, generic PCR, which means most cases, even when they're confirmed, actually only have a generic orthopox virus test. But now There's new protocols being put in place to also include clade-specific testing. But again, if those tests are coming from Kamituga, then those clade-specific tests will be negative. And so WHO is making recommendations to member states around the world to verify their diagnostic algorithms. If they're using only clade-specific testing, then perhaps they will be missing a diagnosis of a clade 1 exported from Central Africa, which is really not something we want to see. So there are also mutations suggestive of like APOBEC activity, suggesting increased human-to-human transmission. This was shown in the global outbreak for clade 2b, and now we have early evidence that this may also be occurring in clade 1 during this, specifically the South Kivu outbreak, I believe. This is another way of showing some of the data, but more by clade proportion, and I illustrated this on the global map earlier. So from 1958 onwards, 58 was the year the monkeypox virus was first discovered, and in monkeys in Copenhagen, in laboratory research animals. And you see that the clade 2A dominated the early decades. Then there was a 2003 outbreak in green in the United States of America, followed by a few more specimens. And then in the pink is obviously the clade 1, which has dominated from Central Africa until as you see in 2017, the Nigeria outbreak began, and we're seeing predominantly clade 2B globally, but very clearly there in 2024, we have a reappearance of a significant proportion of clade 1, again, relative to the sequencing that are available online. And I do want to thank my colleague, Lorenzo Subisi from WHO for this work that we're seeing. Whoops, I didn't realize that I had all these arrows here. Okay, so in summary, we have a continuing rise in reported cases and deaths in the Democratic Republic of the Congo. 1 in 5 health zones are already reporting cases in this year alone, with more than 4,500 cases. Around 10 or 12% of these are laboratory confirmed, and nationally in 2024, the test positive rate is more than 75%. There are co-infections now being reported and confirmed. And as I mentioned earlier, in Equateur, the outbreak focus moves to new health zones. We don't really understand the epidemiology or the forces that are driving that. The role of zoonotic transmission remains not well understood at all, especially in the face of increasing human-to-human transmission. And in Kamituga, South Kivu, the outbreak due to sexual transmission is ongoing. This clearly represents some health security risks, both for the country, but also for the region and the world, given the global immunity gap since smallpox eradication and cessation of vaccination in most countries over 45 years ago, which means anyone under the age of 45 or 50 will be, if they haven't already received mpox vaccines, smallpox vaccines, or had mpox in the last 2 years, will be susceptible to any new outbreak. Studies in animals are continuing. DRC has recently reported positive specimens of specimens positive for monkeypox virus from bats, and this has never been reported before. So we're learning something new every day. And tomorrow we begin an interministerial mpox meeting for the African region for the next 3 days, 2 days of scientific meeting and a ministerial meeting on Saturday to continue to address these issues. So what does all of this mean for public health and response? There's just 2 slides here and then we're done. And that is that clearly there's an impact on epidemiology and transmission dynamics. There are differences in disease severity and fatality, particularly in endemic areas where children are most affected. Mpox has now been recognized as a sexually transmissible disease, and it often co-occurs with HIV and STI, and that can occur in any context. There's possibly enhanced human-to-human transmission for clade 2B, yes, and also suspected for clade 1, an impact on transmissibility, and of course, the reverse impact on enhanced transmission due to continuing viral evolution. For diagnostics, the gene deletions I've mentioned are leading to diagnostic failure of clade 1 specific PCR. This is leading to assessment and review of diagnostic protocols for which recommendations may differ by context, and the need for sequencing for confirmation in some circumstances. And WHO is updating.
Last minute.
Yep. So these are the last 3 points on the public health impact. The performance of therapeutics is affected. There is early evidence of resistance to antiviral agent tecovirimat that was seen in the global outbreak. And performance of vaccines may also be affected by the mechanisms there, and also by changes in modes of transmission. It is currently unknown if vaccine effectiveness will differ by clade, but at the moment, we don't have anything to suggest that. And I mentioned the continuing studies. This is the new global framework for strategic response to mpox and to enhance control and achieve sustained elimination in areas where that's possible, and to continue research and access to countermeasures. That's it. Thank you.
My PhD, I was doing study of a virus called PRRS virus. It stands for porcine respiratory and reproductive and respiratory syndrome viruses, and it is under Nidoviride, outbreak is a positive sense single strain RNA virus that significantly impact pigs. The classification is mostly by the GP5 gene. The naming system is absolutely horrible. We use the cultural, the cell line names. We also use the geographic names, and we also used farm names that discovered the farm. So when I was doing my PhD, I proposed to do a whole genome classification. Of course, I failed as a PhD student. So, and I realized that, yes, I can contribute to this workshop by complying. No, I'm kidding. I guess at the human side, it's doing way much better job than the animal side. But anyway, so I think I represent the state public health realm, and I think it's great for me to talk about what is our current infrastructure, at least. So first of all, we're a part of the state government. We're not part of CDC. We're not part of NIAID or NIH. So we're under the Minnesota Department of Health. So there's Commissioner of Health, and they divided into different bureaus. And the Public Health Laboratory and Infectious Control and Epidemiology are all under Health Protection Bureau. And here on the picture, you will have our public health lab building. And you may wonder, like, why we're showing a bridge out there. I think our CDC colleague mentioned about communicating genomic information challenges with our epidemiologists. There's language differences. Now, this is how we solve the language barrier to build a literal bridge, because no one really wants to get out of the lab building when it's sub-zero outside during the winter time. So within the public health laboratory, we're kind of in this interesting position that we're not clinical laboratories, but we're not research laboratories. But we're doing clinical work, and we're doing the research work. So with a clinical lab, you do diagnostic testing, you do medical management, you focus on individual health. I got sick. I want to see my primary care physicians, and they took my oral swab and they culture it. It focused on me. But for public health, we don't care about you. Well, we do, but we focus on population. We also do diagnostic testing. We do vital diagnostic testing, for example, the TB diagnostic testing. This is something that we test every day, 364 days a year. And we send out results in a timely manner. We also do lots of surveillance and response, and we do communicate public health needs to clinical laboratories. Now, as you can see, our public health lab, we're not just dealing with infectious diseases, all right? We are also dealing with newborn screening. So at least in the United States, every state has a newborn screening programs. And we test, like when you have a baby born, so you do the blood spot and you send to the public health laboratories, and we test, it depends on the states, but a couple of dozens of genetic inherited disease, from cystic fibrosis to, well, don't quote me, I'm not sure about their testing catalog. But yeah, we do test a lot of newborn screening babies. And then there's also the environmental health. So it's organic chemistry, inorganic chemistry, radio surveillance, wastewater, at least they responsible for the wastewater PCR side. And then there was infectious diseases. Now, at least with Minnesota Public Health Lab, it was divided into the microbiology, in biology, in enterics, and of course my laboratory sequencing and bioinformatics. And I just want to add a note here that we do communicate frequently with our clinical labs by using a system called Minnesota Laboratory System. It's an email-based system that can send out notification to more than 160 clinical labs. And this is tremendously helpful during COVID or pre or post-pandemic in terms of communicating our needs to solicit clinical samples for SARS-CoV-2 sequencing. So this is our current infectious disease genomic sequencing program at Minnesota Public Health Lab. As you can see here that in the calendar year of 2023, we already sequenced more than 7,000 non-viral pathogens. And we're only in early April this year, we already have sequenced more than 2,000. So we're not just sequencing SARS-CoV-2 and flu, but we're also dealing a lot with foodborne pathogens like Salmonella, like E. coli, like Listeria, or Campylobacter. We're also dealing heavily with antimicrobial resistant bugs like Klebsiella pneumoniae, Enterobacter, and of course the most horrific Candida auris. Fortunately, we do not have a lot, but we help with our region labs for the Candida auris identification, sequencing, and outbreak detection. Our SARS-CoV-2 program is pretty heavy. So we sequence, this is like since 2023, we sequence roughly around 7,000 SARS-CoV-2 genome on a calendar year basis. We have launched a flu genomic sequencing program in collaboration with Association Public Health Laboratory and CDC. The goal is to sequence about 800 and 1,000 flu genomes on a yearly basis. And we collaborate with a researcher at University of Edinburgh. University of Edinburgh plays a vital role those days with public health response to start the whole genome sequencing of RSV. So those are some of the quick glance of our genomic surveillance program at the Minnesota Public Health Lab. And I do want to open it up that there's a saying that in the public health realm, you see one public health lab, you see one public health lab. Now, we, Minnesota Public Health, is part of the state government, but that is not the case sometimes. For example, the Nebraska Public Health Lab, they are part of the university. The New York State Public Health Lab is in this weird setting that they're part of their New York State affiliated with a university, so it's like hybrid setting. You have all 50 states have a state public health lab, but for some states, for example, California, they are heavily rely on their county-based public health laboratories as well. So there's San Diego County, there's LA County, and you also have city public health lab for those mega cities like New York City Public Health Labs, Chicago Public Health Labs, and Los Angeles Public Health City Public Health Labs. So it's actually quite complicated. And then we're actually looking at, I wouldn't say disparity, but different level of development in terms of the genomic surveillance. In each state, you have more quote unquote advanced states like Minnesota that we basically have everything, sequencers, access to high performance clusters, genomic epidemiologists, and EPIs that is willing to accept new knowledges like phylogenies. Versus you have certain states that it's literally one man's job to deal with genomic surveillance of bacteria, viruses, parasites, and so on. So, and of course, they have very limited resources in terms of the bioinformatics and genomic epidemiology. So that's kind of like the reason that different states at different stages, and we or under the leadership of CDC that sort of like categorize the states into different regions. We have the workforce development, and bioinformatic regional resource to sort of like help from states to other states with their bioinformatic pipeline development and genomic epidemiology, those kind of things. Now I want to quickly switch gears to SARS-CoV-2. So lots of you are part of Sphere's initiative. In Minnesota, we also established similar concept. We call it Minnesota Surveillance of SARS-CoV-2, MNSOS. That is a collaboration of several large genomic sequencing facilities like my lab and University of Minnesota Genomic Center and the Mayo Clinic. We partnered, like I mentioned before, with all the clinical laboratories throughout the states to coordinate the specimen sharing, sequencing, marry with metadata, and upload to the public repository. And of course, this played a vital role in terms of coordination and communicating with CDC in terms of what we need. This is sort of like our one of the goals for our SARS-CoV-2 genomic surveillance. We're one of the first public health lab in the nation who start SARS-CoV-2 genomic sequencing. We discovered the nation's first Gamma variant and the nation's first community-based or community-transmitted Omicron variant. And I think up to now, we have sequenced nearly 70,000 SARS-CoV genomes. So as you can see, we're pretty, high throughput. As I think at the peak of the SARS COVID, our weekly volume is about 1,000 SARS-CoV-2 genome a week. So, and of course, it's not just us, right? And we also rely on our partners, like our federal partners, federal references labs, and the university and Mayo Clinic to help to boost up the surveillance coverage and share not only with CDC, but also locally. So as you can see that on our MDH website, we also mimicking the current CDC setup to lay out our current variant of concerns or variant of interest. Interestingly, a lot of the local folks that they're not interested with CDC's website in terms of the variant distribution. They're more interested with what's happening in their community, in their city, or in their states. So we actually have a lot of website visit in terms of trying to figure out what's going on in Minnesota locally. And I vaguely remember that back to the story that like being one of the first public health lab start SARS-CoV-2. I vividly remember when we finished the first 3 SARS-CoV-2 sequences, and I was sitting in my office and literally screening at who developed the 7-feature column in order to submit your whole genome to NCBI, and because I couldn't figure it out. I think I ended up spending like 5 hours. I saw. I ended up spending like 4 or 5 hours figuring out the 7-feature columns and submitted to NCBI. And of course, a couple of hours later, I got a mystery call from GISAID asking me to submit it to the GISAID. So, but I mean, things have dramatically changed. For the past couple of years, the submission to NCBI and GISAID has, evolved tremendously and become way much easier. And this is, our feedback from the boots on the ground that, things are getting way much easier, and we're thrilled to see those changes. And this is another usage we have for SARS-CoV-2 whole genome surveillance. And this is just, giving out 2 examples that we use those genomic epidemiology to investigate outbreaks, or as much as we can, until the number of cases is out of our hand, out of control. So on the right-hand side, you will see a live transmission. So this is 2 clusters. The one slightly, I can actually use my mouse here. Give me 1 second. So this cluster is a correction facility outbreak, and then this particular one is a long-term healthcare facility outbreak. It was connected by those 2 dots. And our epidemiological investigation find out that those 2 live in the same household, and one person works for the correction facility, and then the other person works as a caregiver at the long-term healthcare facility. And this is, the very first time that we'll be able to identify outbreaks and visualize it of SARS-CoV-2 in a phylogeny perspective. And then we do have another outbreak that is a riverside outbreak of outdoor concert in Minnesota, summer 2020. And this is basically, I think it's 9 or 10 cases that we'll be able to get the whole genome, and they're literally identical with each other. And so we have the evidence to show that even though, back at that time, people talking about outdoor, but even though, it's outdoor, but when you packed people like a sardine in an outdoor concert, SARS-CoV-2 still transmit pretty swiftly. So this is, another example that we use for SARS-CoV-2 whole genome sequences. So what is the end user experience here? To be honest, at the early pandemic response phase, we feel like there's too many cooks, in the kitchen. We got next strain clade, we got GISID clade, we got pangolin lineages, we got Omicron, and we even have constellation designation. So things definitely is a little bit of like in blurb, and our epidemiologists struggle to what to use to communicate with the public. But I think things quickly got cleared up with all of your effort, which we're very grateful. And then there's ongoing limitation of laboratory information management system handling whole genome sequencing data. So you may know or not know that as a clinical lab, that we have to document the patient information for every single specimen coming to the door. As you can imagine, that as a public health lab pre-pandemic, we only deal with a couple of thousands on a yearly basis, but it dramatically spiked during pandemic. But the current LIMS system is not able to handling phylogenies, and is not able to handling FASTA files, not even mention FASTQ files. So we heavily rely on the clade information or lineage information to serve as the biological results of our whole genome sequencing assay to enter into our LIMS system, and then transmit the data either to our epidemiologists through electronic laboratory reporting, as well as CDC through HL5. I believe that's the mechanism we use. And then it's already 3 minutes left. And then the Spheres provided the first ever direct communication platform between the state and local public health laboratories, federal agencies, and academic partners, and which we're very grateful. And this is, very helpful in terms of communicating our needs, our challenges, and our questions, just like one message away. Switch gears, we're talking about RSV now. And I think we can skip this part because old RSV subtyping method is based on the G proteins. And I think this is like a fresh out of Alvin result out of our recent RSV whole genome sequencing. And as you can see that the G protein based nomenclature is not as granular as whole genome sequencing based, which we're adopting right now. So we're working with a researcher called Danny Maloney to adopt this Arctic style whole genome sequencing of RSV. And this is the third respiratory pathogens that we're implementing in the Minnesota Public Health Lab after SARS-CoV-2 and flu. And I think we can skip this methodology part. So we're using the Goya method, like right now, because we realized that the G protein classification is just not enough. So similar to SARS-CoV-2, we also want to lay out what is, the genetic diversity or genomic diversity of RSV, at least in the realm of Minnesota. Like I said, we're a state government. We're responsible for the Minnesota as a whole. So this is kind of like our RSV landscape as for now. We have sequenced more than 400, and it's about half and half between RSVA and RSVB. We do have a couple of interesting cases of co-infection of RSVA and RSVB, which we're not showing the result here for now. So one thought we have is that whether or not even the current lineage classification is granular or provide enough resolution. This is our the most common ancestor tree, and as you can see, there's actually most recent common ancestor. As you can see that our current RSV classification trace back to all the way back to 2009 and 2005. And when we do the similar classification for our SARS-CoV-2, and I usually just dated back to a couple of weeks ago or a couple of months ago. So this is a slide made by my staff. I'm not sure this is confident to say that WGS lineage types are not specific enough. I should put a question mark there, and I leave this question to the experts in the room. Anyway, so this is some general thought from end user experience. As I said before, you see one public health lab, you see one public health lab, and that triggers us to think about when we do classifications, when we do lineage, when we do clade designation. Think about the tools that bioinformatic tools that we should and we can integrate with those names, and whether or not it's easy enough for the end users to implement and to submit our data. After all, not all the labs are as strong as the public health lab. And of course, we still have the current limitation of LAMP system handling WGS data. It's the same thing with our epidemiology database. So either or not, we have to be really good at our clade designation, or we have to figure out a third database that be able to handle the phylogeny data, phylogenies, and integrate the genomic and epidemiological data on a seamless way. And then during the public health emergency that the state and local public health community really needs to have faster consensus of lineage designation and followed with sustainable updates and timely communication. With that, I would like to thank all of my staff at the Sequencing and Bioinformatics Lab, and of course, the CDC AMD office and our staff B group, State Public Health Lab Bioinformatics, for early communication of SARS-CoV-2 methodology, bioinformatic pipelines, and of course, the BV-BRC workshop organizing committee. Thank you.
So any opening questions from the room to get it started, please?
Yes, just to say, if you can hear me, then I'm online.
Can hear you beautifully. Thank you so much.
Derek Smith from Cambridge, Liliana. I'm always so impressed by NIH's strategic thinking in this sort of thing, and also your connections with BARDA and CDC and FDA, etc. How well do you think you have it covered? I mean, it looks very impressive from what you say. Does it feel like it's a complete picture to you? And do you feel like you have sort of enough investigators in these different areas, or what do you feel like the weaknesses are in what you presented?
Thanks for the question. I would say that as a whole, we have tried to cover as many of the essential bases as possible, right? So I think, some of our programs are, of course, trying to address that. We rely heavily on investigator-initiated grants, applications, and programs that they conduct themselves. So all of you can do that as well. But things that are of concern is we have these burst and sort of exploding amount of interest, and then a lot of waning of interest. So investigators sometimes come into the field, they're really excited, they want to contribute, and then they go back into their normal, whatever the other field is. So there is a challenge of maintaining the level of effort, the level of investment, the level of interest in doing all the work that we are conducting right now, and maintaining investment into the pipeline of the workforce, investigators, et cetera, to keep that going. Because, in times when there isn't a pandemic, people might not be as excited to research, one of the lesser known viruses. So that's where we try to do dedicated initiatives for that purpose. And we're fortunate that we have had dollars to spend on that, but it is a concern. I don't know if that answered your question, but.
Question.
Justin Ball, University of Georgia. Andrew, thank you for that discussion. Really interesting. I'm wondering how, I realize you only linked very few cases in that epi outbreak with the genetic data. I think in the paper it's like two groups or something, right? But you said there was a lot of introduction with no contextual sequences. So how much improved resolution do you think you would get if you actually had sequencing from the Afghanistan population or other places where measles is actually circulating?
Thanks for the question. So two pieces of context for that data. So the tree that you saw did have context computed in it. It was graphically extracted for the sake of clarity. So there is some ongoing collection of whole genomes for measles. It's just not conventionally done in a widespread way across the surveillance networks, and it tends to be done on a more retrospective or research basis until those things become more practical for large groups of laboratories to do it. What was the second question?
How much would it be improved if you had that context?
It would be greatly. It would be greatly improved. The one of the things that I didn't really have time to talk about this morning was the difficulty of getting a whole genome from a measles specimen. We use these oropharyngeal swabs that are collected from the patients and extracted, but many of us are familiar with the difficulty of getting whole genomes from patient specimens, and we have to do all kinds of exotic NGS techniques to get the whole thing. So it's labor intensive and it's expensive, and it's a really tough sell to suggest to hundreds and hundreds of laboratories that you all must do this expensive exotic technique that. If you follow me. So did that answer your question? Yeah. So there is a pretty lengthy supplement in the paper that covers a lot of the context, and I think it might answer some of your questions too.
Okay, we have a question in the room, but did Rosamund want to comment? Go ahead.
Rosamund was going to leave, and so any questions we have for her? I have one, or if someone else has a
first one here.
Yeah, thank you very much for your talk, Rosamund Lewis. Cornelius Roma, University of Basel, Next Strain. Because this is a workshop about subspecies classification, I was particularly interested in your view from kind of in the field, how the subspecies classification, so below the clade level as well, how that is used in your day-to-day work, and whether there are any needs that are kind of unmet or could be met there.
Okay, thanks for the question. I hope I illustrated in the talk that the classifications that we have so far, and even the subclassification of 2A and 2B, has been very helpful in describing the epidemiology of the global outbreak. It's really helpful to know that most cases globally are clade 2B. It's also helpful to know that most of them are lineage B. But however, there were a number of lineage A cases detected in all kinds of places, from India to the Eastern Mediterranean, which tells us that there's more than one outbreak concurrently happening, that there were obviously outbreaks of different strains, which is important because as we're at the opposite end of the spectrum from where the speaker on measles is, we're at the stage where the number of strains and lineages is probably expanding as opposed to reducing. We hope to get to that later in responding to these outbreaks. But seeing what's happening in Kamituga, South Kivu, very concerned about what's happening in the endemic areas because we have very little information from there, both epidemiologically and sequence-wise. So in short, I think it's been helpful to a non-virologist such as myself to be able to describe these outbreaks as you've seen me do it today. And I think my virology colleagues at WHO would say the same, that they're working very hard to help advance this area of work and understanding for this particular family of viruses.
Elliot?
Yeah, Rosamund, this is Elliot Lefkowitz. We've heard over these past 2.5 days now how critical the sequencing and bioinformatics is. And I was wondering, and I'm sure that those capabilities are limited, certainly more than you would like. How limited are they across the world? And how far are we in getting the types of capabilities that we really want to see?
I think we have a ways to go. We are trying to provide additional support to the Institut National de Recherche Biomédicale, INRB, in Kinshasa, exactly on this area of work, to both in terms of engaging partners such as yourselves to create a sequencing working group to provide external and remote support, but also to provide hands-on human resource capacity in the country to support this ongoing work, which is essential as we agreed to understand the evolution of the outbreak. But that is just one country. We have Institut Pasteur lab networks across Africa and also Nigeria CDC and so on. They've all grown in capacity also with the pandemic of COVID-19. So that's been super, super helpful. But it's very fragile and it's very resource intensive. And so having both support in kind, technical support in kind, as well as financial support to actually provide additional support to the countries themselves, not only in terms of human resources, but of course, reagents and that sort of thing. So my sense is it's improving, but fragile.
2 questions online.
First question is for Andrew Beck, coming from SIPID, from NIH. Test data plays a crucial role in the software development to verify the correctness, performance, reliability of the software. With the lack of the data accessibility or the continuity of the data with the new surveillance or the continued surveillance, how would you imagine the software be tested? Not to mention that modeling would require dynamic data.
Well, I think that there's probably two answers to that question, and the first one is very short, and it's that academic developers should use the best development practices that are accessible to their group. So, good unit testing packages, good integration testing packages. Software should be as robust as possible. The second thing that I would say is that I envision future use of reference data sets very quite heavily. There's increasing, in some of the meetings that I sit in, there's increasing discussion about the quality of reference data as it exists now, and I see, especially in high throughput sequencing world, I see a use for verified, standard read sets, for example, that could be used to verify that a pipeline works. Those types of kind of simple things that could be human curated and provided to the community pretty easily, I think. Does that answer the question? Did they say?
That's the user question.
Yeah.
But I'll add to that question, surveillance is critical to acquire new data as these strains evolve. And if we don't have enough data, then modeling becomes a challenge because the lack of the data. So the surveillance is important in that capacity. All right, I'll move on to the second question. This is from Dr. Seen, coming from Rumini Gao. What protocol you were used for the RSV whole genome shotgun? Is it publicly available? What was the average genome completeness you were able to get?
Yes. So we'll be able to obtain the whole genome sequence of RSV. So it is a protocol that's publicly available, and it's developed by a researcher at the University of Edinburgh, Danny Maloney. So I can definitely provide his contact information if anyone is interested. And it's Arctic style. I think it's like a couple of dozens of amplicons that's spread throughout the genome.
So back to the room. Question. Ryan,
NCBI. I guess this is mostly for Andrew and Sean. But earlier in the workshop, there was some discussion of sort of like pangolin style lineages for everything? And hypothetically, if that was doable, is that actually appealing from your guys' perspective? And relatedly, we had a big discussion yesterday about how that relates to more immunological epitope type information. Is that a deep concern from your guys' perspective, or do you just really want names, handles? You don't care too much about them mapping to other attributes?
Well, I've been thinking about this, and I think it's, personal opinion. For the record, I feel like it sort of like as a human being, you have your skins, you have your muscles, and you have your cells. So the whole nomenclature and subspecies can mimic that structure. That when you communicate, to the general public. I do think the current alpha, beta, omicron is excellent. The general public do not need to know the B117.1.3, something like that. But for our researchers and for public health realm or researchers, I think it definitely needs to dive into that granularity for laboratory systems and data entry and data sharing and track all the virulence and its related pathogenicity and transmissibility, those kind of things.
I would add that the situation that I described is one in which we don't particularly care about phenotype. Our virus is monotypic, and so we care about classification to the extent that it's epidemiologically meaningful. So this issue of transmission chain continuity and transmission chain discrimination is really our chief concern, and we actually spend considerable time, in meetings trying to, rectify typical types of hierarchical systems to what we would call continuous transmission patterns, et cetera. So that's our chief concern, really, outside of phenotype.
So that will close this. One quick question.
Yeah, and thanks.
Please.
And it's actually directly related to that. I'm pretty naive about measles, but I was curious if you guys are doing a follow-up of the outbreaks, like the Afghan outbreak, to look at the longer-term effects, like the a loss of immune memory, and is there any association at all with genotype, or do people not know currently?
I'm not aware of any association of a particular immune phenotype with any cladistic structure. There's looking at it. Let's put it right now. Put it that way. But to the extent that we have our contacts and collaborations in that part of the world through the measles and rubella surveillance networks, we collect as much data as we can, but it's not always. Sometimes it's not always reliable from certain parts of the world, or it's not a complete picture. There are biases in our data sets that have to do with sampling and that type of thing.